Mr. Dr. Muhammad Akhyar Farooq, PhD in Chemistry, Professor Associate at Formal Christian College, Lahore, Pakistan. Yeah. Then Muhammad Akhyar Farooq has over 20 years of professional and teaching experience. He does research in nanochemistry and physical chemistry. He has received a gold medal, 10 many international and national awards on his academic and professional achievement. He has published more than 120 papers, 37 US patents, 25 books, presented 118 papers. Yeah. And then his skill and expertise, nanoparticle, nanomaterial, physical chemistry, using surfactant micelles. Nanomaterial for energy, hydrogen bonding, material characterization. And before the webinar start, let me Mr. Adi Darmawan to have a welcoming speech for Mr. Akhyar. Please, Mr. Adi.
Okay, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good morning, everyone. Thank you for to Dr. Muhammad Akhyar Farooq from Pakistan. So we have a webinar. We call it JKSA webinar series on chemistry, which is we have every month, and this is the 70th webinar. We invite a lot of people from UK, from even from Professor Peter Etkin, who write the physical chemistry book, and now we invite someone from Pakistan. I think this is honor for us that we can learn, we can get insight from Professor Akhyar about graphene oxide. Actually, I'm also conducting research in graphene oxide, and we use graphene oxide for desalination. So maybe now we can learn a lot about how to maybe to synthesize and to characterize the graphene oxide. And once again, I would like to appreciate and thank you for the time for the Dr. Akhyar. So this is 1 hour, and I hope that we can get a lot of benefit and from Dr. Akhyar. Thank you.
Okay, thank you, Mr. Adi. That's the welcoming speech from Mr. Adi as the head of chemistry department, Diponegoro University. Okay, without further ado, I think it's time for you, Mr. Muhammad Akhyar Farooq, to deliver your presentation. Okay.
Okay, thank you so much. First of all, I would like to thank the colleagues from Indonesia, and this is really an honor for me to present some of our research we are conducting in Pakistan. And this, I'm going to start with my talk. Can you see my slide over here?
Not yet.
Not yet.
Okay.
Can you see now? Right?
Yeah,
now?
yeah.
Yes.
Okay. Thank you. So I'm going to present some of my research, and this is the synthesis, chemical and biological applications of the metal oxide nanocomposite graphene derivative. And here we will discuss about the two derivatives, which is the graphene oxide and the reduced graphene oxide. I'm thankful for your kind introduction, and this is my brief, some of the national and the international awards and my research profile. So moving to the presentation, this is about the my presentation outline, and today I will discuss one of the most important synthesis of the graphene oxide or the reduced graphene oxide with some of the metal oxide by using the green synthesis. There are a lot of methods we have reported, and these are available in the literature in my profile. But more focusedly today, I will discuss more about the green synthesis. What is the mechanism actually behind this to synthesize the, in case of the metal nanoparticles or the metal oxides nanoparticles, and then if you want to do with the graphene oxide on to reduce graphene oxide, what would be the way? So the first moving to the we are working on many of the applications of the nanomaterials, and these are some of them are mentioned over here, and we are lucky to have 7 US patents on these inventions. So later on, I will just highlight the applications of these metal oxides in this area if I have the time. And first, let me start with the what are the different way for the synthesis of the metal or the metal oxide with the graphene oxide. So actually, we have worked on many of the reaction parameters to optimize the parameter to synthesize the nanomaterials, and these are many of them, and we have worked on all of them. And again, as said before, I will discuss today the green synthesis, and in green we consider the plant and the microbes and the fungi as well. But I will only highlight the plant and the microbes, how to synthesize, and little bit the role of the surfactant on the synthesis of the all these nanocomposite. And we have also worked on different methods. These are very simple methods as a chemist student can use in their labs. And again, this would be my today focus about the green synthesis, because I will be using the plant extract and the microbes as well. So these are some of the nanomaterials we have published already. These are available with my profile in some of the publications. So we started with the simple metal, then metal oxides and metal oxides, binary metal oxide, ternary and the quaternary metal oxides. And right now we are working on the some of the graphene oxide and the reduced graphene oxide with all these different metals nanocomposites. So moving to the chemistry behind the green synthesis. Actually, what is happening when you use the plant to synthesize the nanomaterials or the reduced graphene oxide? So actually, if your target is, let's suppose as a reference, I have mentioned over here, any of the reduced graphene oxide nanocomposite, and the first part is that means how can you synthesize? You can take graphite and then convert into graphene oxide and then reduce graphene oxide. If you want and dope any of the metals oxide, first you have to take the metal precursor, convert into the metal oxide, and after calcination you will get the metal oxides. And the third step would be doping of all these two to get the metal oxide dope, graphene oxide, or the reduced graphene oxide nanocomposite. So actually, when we considered it, the reduction of the graphene oxide to the reduced graphene oxide, or the reduction of the metal ions to the metal nanoparticle, that means if I consider the plant is acting as a reducing agent. So I am just playing with the chemistry, what is happening inside. This is the electrochemistry. That means the plant extract or the microbes are acting as the reducing agents. So if you use as a simple plant extract, then it might be difficult to reduce the graphene oxide or the metals into nanoparticles. And the reason is because this is just the graph is showing that with the change in pH, there is change in the potential of the half cells of the water, or this is having the standard potential is 1.229, and for this half cell is 0. And only thing important over here is when you increase the pH, these reduction potential are going to decrease, and these are at 7 pH. That means when the water is neutral, and these are at the pH 14. So we are using this concept. That means you can change the pH and you can change the potential of even the water, and even any of the plant extract or the extract from the microbes So this is the idea behind this one. Let's suppose this is an example. If you want to have the reaction with the water, and there are the two examples, ruthenium and the iron plus 2. And if you look at the standard reduction potential, these two metal salts having low reduction or the less redox potential values as compared to this. So they are not going to be reduced even in the presence of the water. So let's suppose if I increase the pH, and this is the standard, the pH, the pH 7, that means the water is neutral, then this redox potential is reduced, but still both are having less redox potential values. They are not going to reduce and oxidizing the water. So what happens at pH 14? Now the potential is 0.44, and still now you can reduce the iron, this 0.44 is more as compared to this. You can reduce the ruthenium, but still iron plus 2 cannot be reduced at the pH 14. So this is actually the idea actually we are using by using the plant extract. And now I am going to explain what is the mechanism inside. If your intention to prepare the metal nanoparticles, that means the metal in the zero valent oxidation state. And this is one of the examples. Let's suppose if you want to prepare the iron nanoparticles by using this this metal precursor. And of course, this is one of the example of the plant we are using. And the procedure is simple. That means you can prepare the extract and mix with the any of the metal precursor, and then after some process you will get the metal particles. So what is happening actually, this plant is having some of the nitrophenols which are going to act as a reducing agent. So again, if I'm using the two precursor, so what is the effect of the metal precursor? Even that means you can change the metal precursor, then you can change the chemistry to synthesize the nanomaterials. So let's suppose what is happening over here. These are the two metal, the same that your target is to synthesize the iron nanoparticles by using the plant extract. And this is one of the example. Let's suppose many of the plants are having the ascorbic acid and potential is minus 0.2 to 3 volt. And again, in this case, iron plus 3 is reduced because this is having more redox potential values as compared to this, but you cannot reduce the iron plus 2 by using the ascorbic acid. And this is the similar case. These are again the two half cell as I mentioned before. So now I'm going to use a general equation. This is the redox potential of the half cell of the nitrophenols. And the potential is ranging from 0.3 volt to 0.8 volt in general. So there may be a less or there may be more, but just as a reference, I am using this one. So just if you consider the 0.3 is again less, more than as compared to these two. That means these two metal precursors having less redox potential values, they are not going to reduce by oxidizing the polyphenols present in the salt. So what is the answer of this one? So that means now you can change the potential, and in the similar way I have explained in the previous slide, that means you can change the potential of the nitrophenol by using the different concentration of the sodium hydroxide, or simply you can say by changing the pH, the potential half cell potential of the nitrophenol is changed, and now it will reduce from plus 3 to minus 0.88. Let's suppose this is an example, and now these two half cells are having more redox potential values as compared to minus 0.88, and now you can easily reduce even iron plus 2 and the iron plus 3, and you can convert into the iron nanoparticle. So this is the role of the plant extract to reduce the metals into the metal ions. And we have, as said before, we have synthesized the iron nanoparticle by using many of the plant, and then you can use them for any of the application, the chemical application or the biological applications. And here, this is an example. This is again the extract taken from the plant. And when these are oxidized, the iron is reduced to the iron nanoparticles. And further, if your target is to degrade the dyes, these are going to be again oxidized by reducing this methylene blue into the colorless, that is another form of the methylene blue. So this is one of the mechanism I have mentioned over here. We have established there are the other mechanism, it is the degradation or the dye sensitization of the different dyes present in the nanoparticles. So if you have a look over here, we have got around 97.5% degradation, and we have proved this after recycling the same composite towards the degradation of these different dyes. So again, this is not much difference even after getting the 5 cycle. That means you can use this material up to the 5 cycle and get the again the high activity against the degradation of the dyes. And for the biological application, of course, you can use them for the any of the bacterial strains. And here we are using some of the gram-positive and gram-negative bacteria. And this is going to be perfectly acting as an antibacterial agents against these bacteria. So these are the minimum inhibitory concentration we have calculated. And that means these can be used as the antibiotics if you want to formulate the antibiotics by using the iron nanoparticles. So this is one of the mechanism. Now let's see if this is not always that you can prepare all the metal nanoparticles by using the metal salts. So what is happening? What is the other mechanism? This is our one of the recent publication we have published in this year. Let's suppose if your target is the zinc or the tin or chromium or the cerium, and you want to get the zinc oxide, the zinc nanoparticles, tin nanoparticles, or the cerium nanoparticles. Again, if your target is the plant extract, that means this is having at the standard condition, this is having +3 potential. And look at the potentials of all these metal. Just I have put over here as a reference, all these redox potential are much less as compared to the 0.3. That means you cannot reduce all these metal into the metal nanoparticles by using the plant extract at the standard condition. Although you can modify the pH and then you can look whether what is happening inside the electrochemistry. And these are the example as mentioned before. So you can prepare the iron nanoparticles, but who are zinc or tin or the cerium, you cannot prepare, you will get the metal oxides nanoparticle. So now I am going to actually give you the brief mechanism about what is happening when there is the formation of the metal oxides nanoparticle instead of the metal nanoparticle. So this is one of the example. Let's suppose if you are going to prepare a zinc oxide nanoparticle, if your target is the zinc nanoparticle, but zinc is not reduced by using all these plants. So the other possibility is to have the zinc oxide nanoparticles. So the chemistry is mentioned over here. This is a brief mechanism. These are some of the diacid present in the plant extract. So this is the oxidation of these biomolecules. That means they are simply converted into the anions. And then they have the chelation. That means forming the chelation with the metal ion precursor. And then when you hydrolyze by using the water, that is converted into the zinc hydroxide. And when you calcine or dry, it is converted into the zinc oxide nanoparticles. And these are now going to act as the stabilizing agent. So plant extract over here is acting a stabilizing agent, not a reducing agent. And here we have further explore actually what is the role of the different plants by using the metal or the metal oxides nanoparticles. So here one of the example, again the 3 plants are mentioned over here. We use the different methods actually to check whether what is the role of the sodium hydroxide or what is the role of the pH on the change in the potential of the different metal precursors or the nitrophenols. So we are using just sol gel method just by using the sodium hydroxide. And this is the green synthesis, that means just by using the plant extract. And the third one method is the sol gel plus green synthesis, that means the plant extract plus NaOH. So we have actually proved over here that means by changing the method, you can change the chemistry of the metal or the metal oxides and nanoparticles. And in briefly, actually, this is the raw redox potential values of the neodymium, or the +3 to 0 or +3 to +2. Actually, this is important. And these are the redox potential values of the plant we are using, which are present in our plant extract. And look at the values. Again, this value is much less as compared to the value of the plant extract. That means you cannot reduce the neodymium +3 to neodymium 0 valent oxidation state by using the plant extract. So this is the mechanism again we have established. If it is not reduced to the metal nanoparticles, so there is a formation of the metal oxides. And again, this is the one of the we are using as a gallic acid, which is present in the plant. It is converted into anion, and this is the simple removal of the hydrogen. Second step is the chelation of this one with the neodymium, because this is a positive, this is the negative, joining over here. And then with the hydrolysis, it is converted into neodymium hydroxide. And when you calcine, it is converted in the neodymium oxide nanoparticles. So these are the two mechanisms. Actually, this is our recent one mechanism we have established. Actually, we are exploring what is the role of the metals or polyphenols towards the metal nanoparticles or the metal oxides nanoparticles. So this is just I am skipping some of the slides. So the second important, you can use the microbes, because these are also considered as the green synthesis for towards the metal nanoparticles. Again, the chemistry is the same as I have explained before. If microbes are being used, that means the microbes are acting, the enzymes or somehow the chemicals present in the microbes are going to act as a reducing agent. So we have prepared the silver nanoparticles by using the one of the silver and silver salt, and this is the potential is 0.88. And this is, there are the two mechanisms. One is you can synthesize the nanomaterials inside the bacterial wall or the bacterial stain or the microbe stain, and this is called the intracellular synthesis. And one is the outside the wall, this is called the extracellular synthesis. And extracellular synthesis is more convenient because you can prepare outside the wall, and the nanoparticles do not need to remove out of the wall, so they are present and you can easily use for any of the applications. So here we have optimized. Again, the question is, this is the chemistry, and here you can change the pH or you can change the potential of the microbial strains by changing the pH or by using any of the surfactant or any of the salt or the electrolyte. So they are going to change the pH. And we have actually over here identified that or we have optimized that at 55 degrees centigrade or pH 8, or by using this surfactant and potassium sulfate, this is going to act as a best reducing agent, or you can prepare a good particle with a small size by using all these conditions. So this is the good to work. You can use any of the bacterial stain and you just play with the some of the parameter which are going to influence on the redox potential values, and you can get the different shape and size of the nanomaterials. Again, this is, we have further applied to the biological activity, and by changing the concentration, of course, when the concentration is changed, increase the action, again, all these bacterial stain is enhanced. Now I am going to discuss with the, what is the
Hello.
somehow we are now synthesizing the methods, and this is, and actually the comparison why these two are important. So graphene oxide and the reduced graphene oxide, their carbon to oxygen ratio is somehow important, and their electrical conductivity is different. Geo is insulating to semiconductor, and reduced graphene is having a more conductivity. Surface area of graphene oxide is low and reduced graphene oxide is high, or graphene oxide is a little bit to the hydrophilic, and this is hydrophobic. It depends on the, because graphene oxide have the different functions, and these functions are reduced in the presence of the reduced graphene oxide. And this is about the dispersity. Geo is having more as compared to the RGO. So what is the purpose of this one? That means you can choose which is more appropriate for your application. Somehow graphene oxide is more as compared to the reduced graphene oxide, and these are the actually the selection criteria to select one of the graphene oxide or the reduced graphene oxide. So we are using the graphite, and by using the graphite, and you know, this is a 3-dimension. Actually, you are going to separate all these layers and convert into this single layer, which is the 2-dimensional. And here we are creating some sort of the functional group over the graphene that is called the graphene oxide. So actually, to prepare the graphene first, this is the expensive way. So what we are going to do, we are going to oxidize the graphite, which is a 3-dimension, to convert into the graphene oxide, and then to reduce graphene oxide, which is having the more similar properties as compared to the graphene. So what is the way? Way is very simple. This is just a chemical reaction. You can take a graphite powder, mix with the diacid, and then it is converted, starting with the any of the oxidizing agent. You can use different oxidizing agent. This is oxidation of graphite start and at different temperature, and then you can further complete this oxidation process with the H2O2, and this is converted into the yellow solution. And then when you wash with the HCl, this is converted into the purified graphene oxide, and further you can exfoliate into the graphene sheets. So this is actually the chemical methods, very simple. And what is the mechanism? Actually, in the first stage, in the presence of the sulfuric acid or the KMnO4, this is going to convert into the graphite bisulfate. And then this is the intermediate catalyst. It is being produced by the simple electrochemical reaction, which further oxidize this one process. Actually, look at, this is the graphite. Now the functional groups are created over here. This is the graphite oxide. And when you convert into the single layer, this graphite oxide is actually can be considered as the graphene oxide. So graphene oxide is the single layer of the graphite oxide, that is the graphene oxide. So this is important. Now, if you want to reduce, one of the ways is the chemical synthesis. You can reduce the graphite oxide. So there are the 3 functional groups as present. One is this is the epoxy or hydroxyl or the carbonyl groups, and you can use any of the reducing agent, hydrogen hydrate. You can use these, convert actually all these 3 functional group to the reduced form. So this is the reduction process, and this is in general the whole process I have mentioned over here. Actually, I have explained. You can start first with the graphite, complete this one process, and then you will get the graphene oxide, and further you can convert into the reduced graphene oxide by chemical reduction method or the green synthesis. So I will explain today what is the role of the green synthesis to convert this graphite oxide to the reduced graphene oxide. So let's start with the one of the example. This is just, I'm showing some of the examples. This is the europium oxide reduced graphene oxide using this Ficus carica leaf extract. Or first thing is you must prepare the leaf extract. And you can take the leaves, wash them, drying, and then make powder to have extract in the presence of the water. You can use different solvent, but I prefer the water because water, now you can say that this is the green way or the green synthesis. You should avoid any of the other chemicals. That's why this will be considered as the green method. So there are the two ways. How can you prepare the reduced graphene oxide? By using the green synthesis. One of the ways, because this you have to prepare by the chemical synthesis, graphene oxide. And again, if your target is the europium oxide, you can take any of the europium salt, and we are taking europium nitrate, mixed together in the presence of the plant extract. This is called the one-pot synthesis. It will convert into the europium oxide or the reduced graphene oxide. So this mechanism is similar to what I have explained in case of the neodymium oxide, and the nitrophenol present in the plant are acting as a reducing agent to reduce the graphene oxide to the reduced graphene oxide. And this is another way. This is one part and one easier method. This is the more lengthy process. That means first you have to prepare the neodymium oxide by using the plant extract separately and reduce graphene oxide from the graphene oxide separately. Then by using the sol gel method, you can mix together and you will get the europium oxide of the reduced graphene oxide, the nanocomposite. And we have well characterized, actually I'm skipping some of the characterization technique over here because this may be too lengthy, but I'm going to show you the actually what are the general conclusion or the output out of this synthesis. And as explained, we have used europium oxide or the reduced graphene oxide. This is one-part synthesis. This is prepared separately and then mixed together. So look at the values over here, the size. And these two are important, the band gap and the size is less as compared to by using the one-part synthesis as compared to the way two, and its band gap is also less. That means these are going to be highly active as compared to the other way, and this is shown over here by the degradation percentage. This is 88.5% as compared to the 67.5%. So one-part synthesis is much better as compared to the other way. So these are the conclusion. Actually, you can take these as a reference and you can use in your lab to synthesize the nanomaterials. Let's see another example. Again, it is difficult to prepare the copper nanoparticles. So it will be copper oxide. And the reason is I have already explained in the starting slides. And here we are using another plant, and this is the neem, and your target is copper oxide and the reduced graphene oxide. And again, we have used two methods, again, one pot or the separately synthesizing of these two. And again, we have actually verified that the method is with the one pot synthesis is more active as compared to the other. And this is it is shown by the percentage degradation is in 91% because this is having small particle size, small band gap, small particle size mean this is having large surface area, and small band gap mean this is going to work under the visible region as well. So this is acting as a more efficient catalyst as compared to this one. So this is just, you are just playing with the synthesis method, and you can change the activity or the size of the nanomaterials or their optical properties of the nanomaterials. This is one of another example, silver nanoparticles. Now, silver you can prepare by using simple methods. Again, the reason is silver is having more redox potential values than the nitrophenols, so it can easily be reduced to the silver at zero valent oxidation state. And here we are using this material we have prepared from Moringa oleifera, and we are using as the anti-fouling effect. And actually the purpose of this research work, when you filter the polluted water having some of the bacteria, they are going to block the pores of the membrane. So our target is to prepare some of the nanomaterials which are going to act as the anti-fouling properties. So we have proved over here, and this is the way we have synthesized the nanomaterials. Again, this is the long process, but actually the output out of this research is we are using as the anti-fouling properties, and these are tested against some of the bacterias, gram positive and the gram negative, and their images are given over here. So they are going to act against all these bacteria, so that they are acting as or having the anti-fouling properties. That means now the filtration process is not blocked by the bacteria present in the wastewater. This is another example and another application we have done, and this is the lanthanum oxide, graphene oxide, and the reduced graphene oxide. And this is one of the example I have shown over here. You can apply this material for any of the chemical synthesis. And this is one part synthesis of the imidazole. And the process is, first we have, of course, synthesized in the similar way. And I'm going to show you the application of this one. And by using the one part synthesis, you can take three of these compounds to convert into the imidazole, and this is our catalyst, and you can increase the percent yield up to 86% or 89% when graphene oxide or the reduced graphene oxide. So this is the way how the reduce or graphene oxide or the graphene oxide is going to act as an efficient catalyst against the towards the synthesis of many of the organic compounds. That means now you can use these material as the nanocatalyst towards the synthesis of the organic molecules or many of the chemical synthesis. So briefly, now I am going to discuss over here what is the role of the some of the surfactant. Previously, we worked on many of the surfactants. These are the anionic, cationic, or zirconic surfactants on the metal oxide synthesis. So just here, I'm going to explain what is the role of the all these surfactants. So surfactant may be anionic. This is one of the anionic surfactant. It may be cationic or non-ionic or zirconic. So what is the role of the surfactant to control the particle size of the metals or the metal nanoparticles? So look at the example over here. This is the surfactant molecules. When you immerse in a solution, aqueous solution, at the critical micelle concentration, it will convert into this form, and this is called the micelle. And when if your target is to prepare the gadolinium, that means you can use the gadolinium precursor. This is the anionic surfactant. This is going to interact with the cations. These are the gadolinium cations. And when you add the sodium hydroxide as a source for the metal hydroxides, so these are going to interact with the cations. That means cation or the metal ions are interacted by the micelle and the anion. So actually this is going to influence the nucleation process, and it is going to change the particle size. How? If the solution is well dispersed in the monomers, that means you can get the small particle size. But with this formation of the micelle, look at now, this is one of the micelle, and there are many of the cations are joined together. So coagulation of the nanomaterials are start, and you will get the bigger size. So the output is, our intention is to prepare the nanoparticles by using the CMC of the concentration of the surfactant, which is for the critical micelle concentration. So you must keep up to this. Then you have the well dispersed particle having the small size. This is one of another example, sodium dodecyl sulfate, anionic surfactant. We have prepared, previously we have prepared the neodymium oxide alone by using different plants. And here we are actually doping this neodymium oxide with the graphene oxide, the reduced graphene oxide. So when prepared by using with the graphene oxide or the reduced graphene oxide, the band gap is reduced and its percentage degradation against different dyes is also increased. So this is the actually the role of the surfactant. And again, this is the cerium oxide, reduced graphene oxide. And the purpose of using the cerium oxide is there are the three processes are happening whenever, because they are going to act as a photocatalyst. Photocatalyst mean when you fall the light, the electron from the valence band converted into the conduction band, and this is actually the band gap. Then the process of oxidation and the reduction is a start. But instead of the oxidation and reduction process for the cerium, it is going to create the oxygen valences within the molecules, within the structure. So it is going to enhance the photocatalytic or photo degradation activities of the nanomaterials. So this is the importance of the cerium oxide while using the along with the graphene oxide or the reduced graphene oxide. This is another example, samarium oxide or graphene oxide or the reduced graphene oxide. There are a lot of calculation we have done actually, and one most important parameter I want to discuss. We have calculated the bond length by using the IR data, and these equations are given over here. And look at the when it is a graphene oxide, bond length is more as compared to the reduced graphene oxide. This is the bond length of the SMO, samarium and oxygen bond. And the particle size of the graphene oxide with this samarium is more as compared to this one. So that means we are correlating the bond length with the particle size. If the bond length is small, the particle size is reduced. So this is indirectly how can you correlate one of the spectroscopic techniques with the another one. And here we can also use to degrade the nanomaterials, and this is degrading up to the 100%. This is more perfect. This is the last one example, and here we have used the zwitonic surfactant. And zwitonic surfactant mean both are having the positive and the negative charges. And this is also one of recently published article. And look at the values with the reduced graphene oxide is more as compared to this one. And this is due to the band gap, because the band gap of the reduced graphene oxide is small as compared to the graphene oxide. That's why this is going to act as a efficient catalyst by using the graphene oxide. So lastly, I'm just now, I have a short time, maybe the 15 minutes are left. So I am going to show you the very important applications we are doing in our lab. And these are the 8 applications. This is the filtration, energy, forensic, fertilizers, catalyst, medicine, cosmetics, and the skin cares. So starting with the first one, the filtration. And of course, these are helps us to achieve the sustainable development goals identified by the United Nations. These are mentioned against all these applications. The first is the nanofilter. We have actually, these are in generally I am explaining, and you can use many of the materials, and we have used many of the materials we have synthesized in our lab. And the first is the target is how can you actually to treat the wastewater treatment coming out from the textile industries or the chemical industries or the agriculture industries to make it drinkable, or you can reuse the water. So we have prepared 2 ways. One is the column filter, another one is the membrane filter. And these are published in some of the US patents. You can further get the details. So if you are using a column filter, that means you cannot use directly the nanofilter because it will block the pores. So first you have to use the particles filter, which is going to sort out the large particles. Then you can use the microfilter, ultrafilter, and then nanofilter. Actually, by using the nanofilter, you can also filter out the bacteria and the virus. And this is the most important applications of the nanomaterials. And this process we have explored in the last year. We have prepared many of the combination to prepare the nanofilter, and then we have tested against the degradation of the textile water, and this is going to perfectly acting as the as a active against the degradation of the textile pollutant, and this is going to degrade up to the 100%. And this is also one of our important patent, and you can also prepare membrane. The second one application is the energy, and we are preparing some solid oxide fuel cells which are acting as an efficient energy converter taken from the solar cells. And idea is the sustainable nanotechnology or the nanomaterials. And this is actually, you can use these material as an cathode and anode in any of the cells. And these are going to convert an efficient catalyst either from this side to this side. So this is the actually the I have explained over here. Now this is one of the nanocomposite we have used, and the percent efficiency is calculated. This power density is 100 milliwatt per centimeter square. So this is one of the complicated long metal oxides material. We have converted into the simple oxides by using the ternary metal oxides, and then you can see the power density is 83.27 milliwatt per centimeter square. So actually we are now exploring different the solid oxide fuel cells to act as the power generation. So you can use as the solar cell of the photovoltaic cell. In nanoforensics, actually this is also one of the important application. And your target is how can you, because in the forensic, that means you have the explosive materials, and our target is how can we degrade these explosive material, or how can we use them as the nanopowder for the lifting of the latent fingerprints. So it will help us to investigate the crimes. So we are using some of the nanomaterials. Again, these are the US patents. We have prepared some of the nanopowders. This is the general mechanism. That means this is one of the calcium oxide or aluminum oxide nanoparticles you can prepare. And when these are going to interact with the nitrophenols, which are the explosive compounds, these are converted into the less toxic compounds. And we have verified this mechanism through the GCMS. That means what are the different degradation products out of the nitrophenols. And the second one important in the nanoforensics is the fingerprinting. We have established the mechanism, how they are going to act as an efficient material to lift the latent fingerprint. That means when your surface area is oily or porous surface, then generally the commercial powder are not efficient. So nanopowder will be more efficient towards this. And this is the one of the application. One, it is we have further modified the nanopowder by using the fluorescent dye, and you can further modify by using the magnetic nanomaterial. This is the iron oxide. So this is the simple one example. This is acting as the magnetic material, and you can easily identify the pores and size, the verification of the latent fingerprints. All these are our real pictures. The fourth one application is the nanofertilizers. Again, just winding up my slides. So we have synthesized many of the fertilizers. These are the potash, zinc, or micronutrient fertilizers. These are some of the references I mentioned over here. You can further explore. So idea is how can we prepare a slow release fertilizer. So it is slowly taken up by the plants. So it helps us to reduce the degradation of the leaching of the fertilizers, which is going to pollute the water bed. So idea is we are using the aluminum silicates, and by using the surfactants, when you prepare the nanomaterials, as I have explained before, when you burn, the surfactant is taken out from the materials, it will create the pores, and then you can put some of the urea or the potassium, calcium, ammonium nitrate, or any of the fertilizer inside the pores, and this will slowly release by the time so it is taken up by the plants. We have checked the release up to the 400 hours, up to the 16 days. We have moved to the 1 month. It depends on what is the requirement by the farmers of the crops. We can modify these fertilizers. See, these are the zinc fertilizers. So these are the real picture we have tested on some of the crops. This is the rice crops. And to look at starting from, we have changed the concentration of the fertilizers. And these are the two examples. This is the plant by using the control or the commercial urea. And this is what we have prepared in our lab. And so look at what is the size of the plant by using the nano fertilizer as compared to the commercial fertilizer. So this is now another example, and you can see again, sorry, there is some, okay. So for the nano catalyst, as I explained, you can use in many of the industrial catalysts. One of the application is to convert the styrene to ethyl benzene, and we have used all these nanocomposite. This is very early, one of the publication. We have used tin oxide as a nanocatalyst to convert the styrene to ethyl benzene. That means in many of the chemical industries or many of the chemical synthesis or the organic synthesis, you can use them as a catalyst. And again, this is the imidazole without reduced graphene oxide I have mentioned before. And this is the iron oxide and the cerium oxide. And we have further actually enhanced the conversion efficiency of these materials as compared to without the nanocatalyst. And this is with the reduced graphene oxide. And this is, I have already shown, we have increased up to the 89%. This is the same sensor. And for the nano drugs, we are developing some antibiotics, anti-cancer drugs. So we are testing now actually the radio labeling in one of the nuclear hospital in Pakistan. So actually the purpose is, again, we are developing some slow release drugs. So let's suppose if the doctor will give you the 2 tablets in a day, it is our tablet may be acting as it's sufficient for the 2 days. And this is, we have established the results. This is with the commercial. It is working up to the 6 hours, and then you can further increase the efficiency with the nano drugs up to the 24 or the 48 hours. So this is actually the mechanism. Maybe in some other lectures, if you are interested, maybe I will give you the detailed description on any one of the applications. So nano cosmetics, this is also one of the interesting area right now. Many products are available. You can use them. This is the FDA approved materials. You can use them to cure the skin or to treat the skin, to developing some of the nano cosmetics. And this is the last one application. We have developed some body care products. And again, these are the silver, nano silvers with some the herbal or some. This is a sulfate and paraben free shampoo because the available is carcinogenic. We are replacing with the sulfate and paraben free shampoo and hand sanitizer. There are a lot of other products we have developed in the lab by using all these nano materials. So again, this is our right now available nanomaterials in the form of the publications. And these are some of the publications, the journals in which we have published our work, some of the very good journals across the world. And at the end, I would like to acknowledge my students, of course, my PhD student, MPhil, and the BS students. These are the funding organizations, international or the national funding, which help us to establish the nanomaterials lab in Pakistan. And these are my current research student last year, 2021, and these are recently working with me. These are the PhD, MPhil, or the BS students. So with this, I would like to thank your patience to bear with me to actually to listen this long presentation. So with this, I would like to end and tune to the professor to address any of the questions if you are having. I am available. Thank you so much.
Okay, thank you, Mr. Farooq. That's nice talk, yeah. You related your research by sustainable development goal in United States. From what is it? Okay, I think we start from to the question, yeah.
Yes.
Why oxidation reduction using plant is more effective? This is the first question. Yeah.
Sorry, can I read or can you repeat the question?
I think it is up here on the screen.
Okay. I just thank you. Why oxidation reduction using plant is more effective? So this is might not be most effective. We are just using the plant actually to moving toward the eco-friendly or environmentally friendly senses. Even some of the reducing agent, chemically reducing agent like sodium borohydrate or something else, they are considered as the more effective. They can reduce the metal ions to the metal, but the purpose is not over here is the plants are more efficient or the more effective, but the reason is we are moving from the chemical senses towards the green senses. So this is not the question of the effectiveness, this is the question of the environmentally friendly senses, and that is called the green senses.
Okay, can continue next.
Okay, what are the factors that influence the effectiveness of green senses except the reduction potential? Okay, the other factors are linked with the redox potential values. It may be a pH, it may be a temperature, it may be a feed rate. If you remember in one of my slides, we have studied the different factors, and it is starting from the feed rate, the way you are adding some of the base to the metal ions, the different solvent you can use, different surfactant you can use, or that temperature you can change, or the pH you can change. These are the different way or the different factor which are going to influence on the green senses. So actually indirectly they should influence on the redox potential values. If they're not influencing the redox potential values, that means actually we are neglecting the electrochemical reaction. So here there are the factors as I have mentioned, but directly or indirectly they are going to influence the redox potential values, and this is going to further influence on the senses of the metal ions to the metal nanoparticles. Yes.
Okay, next.
Okay, how do we know what substances that are responsible to the reduction of metal ions if we use green methods such as plant and microbes for preparation of the metal nanoparticles? Okay, actually what we are doing in our labs, we go through the literature and identify because if you start searching the plant which are having the more nitrophenols, then it will be more lengthy process. So actually the purpose is you can select that plant or microbes which are having high concentration of the nitrophenols. So the best idea is you can go through the literature and search such plant which are having more nitrophenols, and you should use these plant towards the reduction of the metal ions to the metal nanoparticles, and similar is the case for the microbes. So this is the idea, that means how and substance which are responsible are the nitrophenols, and this is the way I have explained to search through the literature. Otherwise, you can also, if the plant is new, you can also phenolic compounds, flavonoid, or the other things. Organic chemists much know better to calculate all these things, and you can also calculate and then further you can apply. But this way would be a more long process to work the synthesis of the nanomaterials.
Okay, we can then continue.
Okay, in this direction, contest which is better, using plant or using, okay, as I have explained, if you understand actually what is the purpose of using plant and the other oxidizing agent, and then I think you would be better to understand what is the purpose of using plant or what is the purpose of using any of the reducing agent. So here, as I have said before, the reducing agent, this or the other reducing agent might be more efficient towards the reduction of the metal to the metal nanoparticles. But the question is, because there are the side products which are remain present in the during the synthesis. So by using the plant extract, our purpose is to avoid such type of the oxidizing or the reducing agent. So this is only the difference between the plant's extract or the reducing agent. Otherwise, if you compare, the chemically reducing agent are more efficient towards the reduction of the metal ions, but they are not eco-friendly. Plant are more eco-friendly as compared to the other oxidizing agent. So this is the actually the difference. You have to decide what is your purpose. You can use accordingly.
Okay.
Okay, what is the metal oxide? Where is the metal oxide located in graphene oxide structure? How is the bond between metal oxide and the graphene oxide? So I think this is the work which is going on. Actually, what process we have identified right now, because it needs a lot of the mechanism. Actually, we have studied that this is only the adsorption process. And if there is a graphene oxide or the reduced graphene oxide, your metal is adsorbed on the surface and it will give you the composite, as we are going to prepare the other sol-gel, other composite by using the sol-gel method. So here right now we have not identified actually which part of the carbon is going or the other functional is going to replace with the metal ions. So this is still the question which has to be searched out.
Okay, next.
About the role of plant extract when GO becomes RGO, what is the variable? What is the variable should be emphasized? The extract is crude compound. It is possible to get the same result when we synthesize twice. So the first, I think, what I can understand, what is the variable result? What should be the difference between these two? And actually, I have mentioned during this slide, it depends on what is your purpose or the applications. Let's suppose, again, just one of the parameter I am going to highlight. This graphene oxide is having high band gap as compared to the RGO. So if you want to work in the visible region, then RGO is more efficient as compared to the GO. This is the question to the first part. So the second is, of course, the extract is a crude material or the crude extract. It is having many of the nitrophenols present. And of course, you can, when you prepare by using the plant extract, that means the only thing is you have to take the similar amount of the plant. That means, let's suppose if you are taking, we have established 1:4 ratio between the metal and the plant weight. So 4-time plant is more as compared to the weight of the metal precursor. So if you use this combination, then you can repeat the result. And if you vary, of course, if you vary the concentration of the metal or the plant extract, then of course there is the possibility to change the results of the nanomaterials. Yes.
Okay, next.
So what is the role of graphene? What is the role of graphene in copper oxide, or sorry, what is the role of graphene? Copper oxide graphene, is it an anti-buffer layer or graphene doped copper oxide? As explained before, this is only right now what we have discovered. This is adsorbed on the surface of the graphene oxide. This is not the graphene we have prepared. This is the graphene oxide or the reduced graphene oxide. So this copper oxide in the one-part synthesis or the other way, this is just adsorbed on the surface of the graphene oxide or the reduced graphene oxide. So this is the, I think, the answer of this.
Okay. Next.
Okay, what do you mean about metal oxide doped graphene? Is it same with modification of the metal oxide outside the graphene, or substitution of carbon in graphene structure by metal cations from the metal oxide? This is a good question, and the answer is, whenever you use the word doped, that means you have rightly said in the second line, third line, one of the graphene, one of the carbon is substituted with the metals. Then we use the word the doping of the metals with the carbon, and you can use the word the metal oxide doped graphene oxide. Again, this is not the graphene we have prepared, this is the graphene oxide. So again, this is just by the question. If it is replaced, we call it the doping. Otherwise, this is just called the adsorb. So as I have said, both processes are going on, but we have not discovered, this is reported in the literatures, maybe somehow there is the replacement of the the carbon with the metal oxides, or some of the functional present on the graphene oxide with the copper oxide, then you can use this term. This is the doping of the metal oxide on the graphene oxide.
Okay, next.
Okay, what is the minimum potential reduction value that is required plant in order to do green synthesis? So it is not the reduction potential of the plant you have to decide. The first you have to decide what is the potential of the metal precursor. As I have explained, if your target is the silver or the iron, then you can easily synthesize the nano particles by using the green synthesis, because the reduction potential of the polyphenols present in the green synthesis, it is ranging from 0.3 to 1 point something, as I have explained. And if you prepare by using the sodium hydroxide, then the potential is reduced. So it depends what type of how many nitrophenols are there. So actually results will give you the indication. This is the XRD will give you the indication whether you have prepared the metal nanoparticles or the metal oxides nanoparticles. But you can take the idea prior to the synthesis, as I have explained. Maybe you can watch again the video I have discussed in detail. How can you prepare the metal of the metal oxide nanoparticle by using the green synthesis?
Okay. Next. What's strength metal?
How strength of the metal oxide and the green graphene surface interaction, and how to strengthen it. Again, this is about the mechanism. As I have said, we have not just discovered this mechanism, but of course, it depends what is the we call it this is the isoelectric point. Actually, whenever you adsorb any of the metal oxide onto the other it depends on the isoelectric point, and at isoelectric point, that means at this is going to act as a neutral. So we consider this, and then you can change the pH by increasing or by decreasing. Then you can absorb any of the metal oxide on the graphene oxide, and then further you can explore the properties. And this is actually we have done with the metal oxides, not specifically with the graphene oxide. So of course it will influence on the interaction with the metal oxide and the graphene oxide, and isoelectric point is the point to be considered.
Okay, the next question. Which one is?
Which one is better membrane? Is better for membrane zinc oxide, graphene, or zinc oxide silicon dioxide? Actually, I don't know the purpose of this, or maybe you are going to prepare a membrane for the filtration, or it may be for the anti-fouling properties, as I have discussed. Generally, we have used the, this is a polyethylene glycol or polyvinyl alcohol. You can use all these membrane to modify with the nanomaterials, which you have mentioned over here. And there are a lot of other polymers as there. You can use that.
Okay. How to control?
How to control the size of nanoparticle, which is synthesized using plant extract. And again, this I have explained during the presentation. One of the way is by using the different amount of the plant and by using the different plants. And I have explained in the lab, when we are using different plants, having different combination or different proportion of the nitrophenols, we have prepared different sizes of the nanoparticles. So this is the answer of this.
Okay, next question.
Will the nanoparticles have the same properties when it is synthesized using green route compared to the conventional method, chemical reagents such as using precipitation and calcination? Okay, the property actually, of course, the properties should be different. If there are the same properties, in general, if I consider, let's suppose if one of the, you have prepared the nanocomposite, let's suppose the zinc oxide or the zinc or the iron oxide or the iron nanoparticles by using both method, and the both will act as the photocatalytic degradation of the dyes. The theme is the same, but of course within the degradation, the properties of the both will be different, and this depends on the size, which method you are using to control the size, and what we have identified over here. When you use the both of the combination sol-gel and the green synthesis, particle size is less, and then your activity or the photocatalytic activity will be more, and this is going to influence on the properties as well.
Okay, next.
How to produce nanoparticles of your product. And this I have, actually I have not discussed the detail due to the time, but at the start I have mentioned we can use the sol-gel method, we can use the precipitation method, we have used the hydrothermal method, we have used the wet oxidation method. There are a lot of methods, green synthesis by using the microbes. So all these methods you can use to produce the nanoparticles. Next. Is there any specific criteria of plants which can be applied for the nanoparticle synthesis using green route? Again, this is the same question I have addressed before. Of course, this is the criteria. You should select a plant which is having more nitrophenols, and this you can check through the literature. Okay, in the process of synthesizing metal nanoparticle with plants, also need a stabilizer. I don't think if you want to add, you can add, you can explore this one property, but the plant or the nitrophenols itself are acting as a stabilizing agent. They are the stabilizer. That's why we are using them toward the reduction, or they are also acting as the stabilizing agent. There is no need for the additional stabilizer.
Okay, next.
Okay, if we want to use natural surfactant, please give us your suggestion what the important points of surfactant parameter such as CMC. How about if we are using the chemical synthetic surfactant? So as per my knowledge or what we have done in our lab, it is natural or it is synthetic. Only important is the critical micelle concentration. And if you want to prepare the nanomaterials with the small size, you must use the critical micelle concentration, not more than the critical micelle concentration. And if this is not known, this you can get from the literature. And if it is not known, this is also very easy. You can use the conductometry, you can use the pH metry, depending on the nature of the surfactant, and you can calculate the CMC.
Okay, I think it's enough the question. Once again, thank you for Dr. Muhammad Akhyar Farouk for your nice
Thank you, sir.
presentation, comprehensive discussion. Okay, that's nice. Maybe that will be useful for us as the participants. Dan terima kasih pada Bapak Ibu mahasiswa yang telah ikut di webinar ini. Okay, thank you, Mr.
Okay, thank you.
Farouk.
Thank you, sir. Okay,
Have
thank
a
you
nice
so much.
day. Okay. Assalamualaikum
Assalamualaikum.
Waalaikumsalam.
Foto, Pak.
Foto. Ya Pak Adi yang bikin. May we have a photograph, Mr. Farouk?
Yes.
This is the photo session. Can you turn on your camera?
Yes, I have turned on.
Okay.
But it says dark, right?
Is it still dark? Can
Yeah,
you?
yeah. Actually, I'm sitting in the office, and there is only the two lights are present over here. So,
Because I can't see your
okay.
Face.
can't see your space. Can you switch on your webcam like at the beginning?
Let me.
Okay.
I don't know. Maybe you can take my this picture.
Okay. Sorry. So 1, 2, 3. Okay. The next slide. 1, 2, 3. And the last one is 1, 2, 3. Okay. Thank you very much, Dr. Akhyar, for your nice presentation. It was very amazing. So I got to give to Pak Gunawan. Pak Gunawan.
Okay.
Thank you.
I think it's time up for the audience. Thank you, Dr. Muhammad Akhyar Farouk, once again.
Goodbye.
I will be very interesting discussion here. Have a nice day. See you next webinar.
Thank you.
Thank you very
Thank
much.
you, Dr.
Thank
Akhyar
you.
Farouk, for your time and also sharing knowledge here.
Oke, for semuanya terima kasih Bapak Ibu.
Terima kasih Bapak Ibu sudah hadir.
Terima kasih.